questions, jump in. Okay, so this is going to be about gene regulation, all right? So we're going to talk today about how genes are controlled. We're going to focus on one really big idea, but there's going to be a few other smaller ideas to consider when discussing gene regulation, all right? The big idea is that genes are controlled by proteins. These proteins are called transcription factors, which kind of makes sense because if you remember in gene, when you copy a gene, that's called transcription. So a transcription factor is a protein that actually regulates transcription. They're also called regulatory proteins. You're going to come across this term also, okay? These are, the big idea is that these proteins regulate in some way transcription, the ability to copy a gene and use it. So here are three examples of, you see these little purple things, these are proteins that are attaching to DNA. Here's an example of one, okay? You can see it's attaching to DNA. If you remember from your biochemistry, proteins can have these regions called domains. And what all transcription factors have in common is they have a domain called a DNA binding domain. You remember what a DNA binding domain is? It's exactly what it sounds like. It's a part of the protein that actually attaches to DNA, and you can see it here. So the way they do that is the domain recognizes sequences on the DNA. Here are some examples. So here are some transcription factors or regulatory proteins that were found in, for the most part, fruit flies. I think these might have all been found in fruit flies. But like, for example, this one here called abdominal B, you can see it recognizes particular sequences on the DNA strand. So if it sees, like, for example, the sequence TAA, TTT, GCAT, then it attaches to the DNA. It also recognizes another sequence. You can see here, TCAA, TTAAT. So these are just examples of different regulatory proteins or transcription factors and the sequences that their DNA binding domains recognize. Because the big idea is when they attach to the DNA, they are going to somehow regulate the transcription, which simply means one of two things. Either they're going to turn the gene on or turn it off. That's what's going to happen, okay? So we're going to actually take a look at how this works. What we're going to look at is an example in bacteria, but basically the way it works in bacteria is pretty much the same way it works in eukaryotes. We're going to look at prokaryotic regulation, okay, which is going to be, I mean, overall it's simpler than eukaryotes because they don't have, bacteria don't have things like hormones to also regulate genes. If you remember, bacteria don't splice their mRNA. So it's a little bit different what bacteria do than what eukaryotes do, but the big idea is still the same, okay? And then the way they control their genes is going to be the same way that we do it. So before we begin, just some quick terminology about genes. Genes that are always being expressed, they're called constitutive genes. That's the first term right up here, okay? These are genes that are always on. So for example, some of the enzymes in glycolysis, they're always being expressed, which means those proteins are always being made. An inducible gene is a gene that is normally off, but can be switched on. And a repressible gene is the exact opposite. It's normally on, but can be switched off. So we're going to focus on the ones that we can switch on and off, the inducible and repressible ones. We're going to look at a model to help understand this. The model is found in bacteria. This is called an Operon model. And the main difference is that in an Operon model, if you remember from transcription, the role of the promoter region is to allow RNA polymerase to attach to that site and then begin transcription, right? In eukaryotes, one promoter region controls one gene. So one promoter region controls one gene, but in prokaryotes, an operon is where you have one promoter region controlling possibly many genes. That's really the difference. You can see that actually here. So in this example, you have a promoter region and you have 3 genes that are actually controlled by the promoter region. You can see here, these 3 genes are called Lac Z, Lac Y, and Lac A. So in bacteria, if it's an Operon model, it's one promoter region controls several genes. But again, in eukaryotes, what we see is one promoter controls one gene, okay? But that's not really important. What's really important is the way it works, which is going to be the same whether it's in prokaryotes or eukaryotes. So the organization might be a little bit different, but the method of which this is controlled is the same. So I'm going to show you 3 examples, okay? 3 examples of this and how proteins regulate transcription. First one is the Lac Operon. So this is an operon that is inducible, which means it is normally off, and we can switch it on. Okay, so it's inducible. And it has 3 genes that are under the control of the promoter region. The third one is one that is still kind of a bit of a mystery as to what it does, but the first 2 are well known. The first one is the gene for an enzyme that breaks down lactose, okay? And the second is a gene that actually brings in lactose. And if you look at the name, it's called permease, so it actually allows the lactose to enter the cell of the bacteria. So if you imagine this is the cell of the bacteria, the permease is basically a doorway that allows the lactose to enter into the cell. So let's take a look at how the Lac Operon works, okay? So in this picture, what you see is something called a repressor protein, okay, which is right over here. Now, the repressor protein is an example of one of those regulatory proteins. It has a DNA binding domain. You can see that's actually attached to DNA. And if you notice where it actually sits in a region called the operator, which is in between the promoter region and the genes that we want to copy. So if RNA polymerase were to try and read this gene and transcribe it, it wouldn't be able to, because this repressor protein is actually physically in the way. So this gene is off currently. Now, we need to switch it on. So how does it get switched on? Well, if you remember from enzymes, a topic on enzymes, proteins have these regions called allosteric sites. So if this were to be considered the active site, this is the allosteric site, okay? Now, that allosteric site is a region where we can have something attached to it that would regulate it. It would either switch it on or switch it off. In this case, what switches it off is lactose. The presence of lactose right here actually binds to the allosteric site and changes the shape of the protein. You saw this before when we talked about enzyme regulation. This is no different. Now, if you remember from the topic that form and function are connected, you can see that if we change the shape of the protein, then it no longer attaches to the DNA, falls off. Now it's out of the way, which means the RNA polymerase has free access to the genes, which now means basically that the gene has been switched on. So that RNA polymerase then transcribes the genes for the enzyme to break down the lactose, the enzyme to bring in the lactose, and this relatively unknown enzyme. Now, you may have realized there's a bit of a issue that comes up because one of the enzymes that is made during this process when the gene is switched on is the doorway to bring in the lactose. So how does the lactose get in the first place to turn the enzyme off if this is the doorway to bring it in? Turns out that if you go back to the cell, this enzyme, the permease, the protein, the permease protein that brings in the lactose, is only one of two transporters. There's actually a second transporter that's already sitting in the cell membrane that allows the lactose to enter. This transporter is then made once the lactose enters. The transporter is made. So the reason why we make more transporters to bring in the lactose is because I guess you could say that the cells are greedy, and the more doorways we can put in the cell membrane, right, then the more lactose the cell can gain. That's not really that important that you know that. What's really important is the way this works, right? The idea is you have a regulatory protein attached to the DNA. You can see that where it's sitting, it's blocking the RNA polymerase. And if we can remove it, that means the gene can be switched on. That's the big idea. Okay, questions? Any questions so far? No questions? Okay, so let me show you another example that's going to look very, very similar. I'm going to scroll down to something called the trip operon. Okay, here's the trip operon. Now, this is an example where the gene is repressible. So the genes are on, but we can switch it off. So you can see in this picture here that the RNA polymerase there's nothing in the way. So in this slide, you can see that the RNA polymerase, which is over here, can copy this. In this case, the trip operon is 5 genes. If you recall, when we did biochemistry, we talked about a feedback loop where we make one amino acid and convert it into another amino acid. And at each step, there's an enzyme that makes the next product. And then when you make your product, you have a feedback effect, right? I don't know if you remember that, but what you're seeing in this slide is the production of 5 different enzymes. So each of these enzymes, so the enzyme we'll call E, D, C, B, and A, corresponds to one of those steps in the pathway. So if we're making tryptophan, that means that it requires 5 steps. So there will be step 1, 2, 3, 4, 5. Each of those steps is controlled by an enzyme. So at step 1, we make something, then at step 2, we make something else. And then at step 3, we make something else. Step 4, we make something else. Step 5, we eventually make tryptophan. And then tryptophan would feed back and turn off enzyme 1. Remember, that's your feedback loop. So here's my repressor protein right over here. You'll notice that it's not attached to the DNA, right? Now you'll notice the way it's drawn. It has coincidentally the same shape as this tryptophan molecule. And the reason for that is because when you make enough tryptophan, you don't want to make any more. So what happens then is the tryptophan, you can see, acts as the allosteric regulator, which means it will bind to the allosteric site. You can see it here, which then changes the shape of my protein. Now my repressor protein becomes active. And you can see that it binds again in that same region that will block the RNA polymerase from being able to now transcribe the genes to make tryptophan. So this is, I mean, if you compare this to the Lac Operon, you can see it's the same idea, right? It's the idea is you have this protein that either will attach to the DNA or not. If in both cases, if they attach to the DNA, so you can see here in both cases, when it's attached to the DNA in the operator region, there's no transcription. You can see that here, and you can see it here. There's no transcription. And in both cases, if the repressor protein is removed, then what we have is we have transcription is occurring, right? You can see that this is a big idea of how proteins regulate transcription. I'll show you basically a third example, but the third example is actually just a follow-up to the Lac Operon, okay? And let me go back to actually an idea which kind of should be logical. If you think about the trip operon, we want to turn this off when we have enough tryptophan. I mean, here we're making tryptophan, that's the idea. And then when we have enough, we need to shut it off. In the Lac Operon, we don't want to make the enzymes to break down lactose unless there is lactose, otherwise it would be a waste of time. So when do we make like the enzyme to break down the lactose? Well, that's when lactose is present, which removes the repressor protein. Okay, so there's a logic to the way these operons, or basically gene regulation, works. Now, going back to the Lac Operon, the Lac Operon is again a set of genes that are, when they're switched on, they make enzymes to break down lactose, basically bring it in and break it down, because the lactose is a carbohydrate, from your biochemistry. And if you remember from energetics, we can convert carbohydrates into ATP. The thing is that lactose is a second choice in terms of energy production. The number one choice for energy production is, if you recall, because we focused on it, is glucose. Lactose, in this case, comes in second. Okay, so what happens when you have both available to a cell, glucose and lactose? It turns out that the cell focuses on glucose. So what does that mean? So when lactose is present, we know that the lactose binds to the repressor protein, right here, and knocks it off. So we have transcription. Okay, and you can see that here. The transcription is nothing blocking RNA polymerase. So that means we can transcribe these genes. But I made a comment before that basically the way this works is you have proteins, they attach to the DNA, and they would do one of two things. They either turn the gene on or they turn it off. Now, truthfully, that's a little bit of a lie, because we can add a third possibility, or a third thing to consider. Think of it more like a dimmer switch. Okay? So what a dimmer switch is it allow you to turn lights or something on. Let's think about lights, okay? And we can turn them off. But it also regulates how bright the lights are. So what does that really mean? That means, in terms of transcription, we can turn genes on, we can turn them off. But if we think of them as a dimmer switch, we can, if we turn them on, we can increase the transcription rate. Now, why would you want to do this? Okay, so if glucose is your number one choice and lactose is present, then the cell is going to focus primarily on the glucose. So it will break down the lactose, but it will break it down kind of slowly. It's like the lights are on low. The genes are on, but they're being transcribed slowly. Okay, and why? It's because glucose is present, and that's the top priority for the cell. What if glucose wasn't present? What if glucose was absent? Then what the cell has to do is focus on the lactose. It's got to be better at breaking it down. So what the cell does then is the cell increases the transcription rate of the genes to bring in and break down the lactose. In other words, it raises the dimmer switch. And how it does this is there's a protein, and that protein is called CAP. You can see it right here, okay? Now, you can see it says that CAP is inactive. CAP is inactive basically in a simple way when glucose is present. Okay, so when glucose is present, CAP is active. Sorry, inactive. When glucose is not present, then there is a molecule that increases in concentration called CAMP, which you can see right here. And what CAMP does is it allows CAP to form into its active state. And when it forms in its active state, it can then bind to the DNA. So again, it only does this, it only becomes active when there is no glucose. Because when there's no glucose, there's high levels of CAMP. And the CAMP allows the CAP to form into its active configuration. So now look what we have. We have a molecule that's active, but look where it binds. It doesn't bind in the way of the RNA polymerase, because that would be in the operator region. It binds in an area that is out of the way. But what it does when it binds is it actually helps to open the DNA. Because the faster the DNA opens, the easier it is to open up the DNA, then the faster we can copy down and transcribe and translate these genes to make the enzymes required to break down lactose. Okay, so these are 3 examples of regulatory proteins that you can clearly see. They're proteins that are attached to the DNA, and they do one of 2 things. They either turn the gene on or they turn it off. Okay. Any questions so far about this? Anything in the chat? Nothing? You girls still there?
Yes.
Yes, sir.
Okay. No questions about this?
No.
Okay. All right. So then let me show you like an idea, because I think I might have actually showed you this before. So what we're talking about here is the regulation of genes, right? So that means talking about genes being turned off or turned on, but we can add a bit more to that. We can also talk about if the gene is turned on, you know, like how much of it, like is it turned on loudly, thinking about like in terms of sound, or like a dimmer switch, right? The rate of transcription, we can think of it as if it's being transcribed really, really quickly or really, really slowly. And the big idea is that these are proteins that control this process. And this was discovered in E. coli bacteria by 2 scientists named, you can see here, Jacob and Monod. And when they discovered how this works, and I think it was a Lac Operon they were working on, they said this. They said, what is true for E. coli is also true for the elephant. So in other words, what we just talked about and how this process works, how regulation is controlled by these proteins, these regulatory proteins, is true in bacteria, but it's true in all things. Okay, that's what the statement means. And it also allows us to think of genes, and we talked about this a little bit last year. You can think of genes as like switches. Think of them as switches, because really that's what these things are, right? I mean, these are ways to switch on and off genes. And if we think of them as switches, we can actually maybe understand a little bit more about certain types of mutations, because our next topic will be on mutations. And one of the things that will come up is we're gonna focus more on like mutations inside a protein. But there are in humans, probably most mutations are outside proteins, because only a small percentage of our genome actually codes for protein. And if you think about mutations that are outside the coding regions, the parts that give you protein, these are mutations that could possibly affect the switch. So for example, if I go back to here, okay? So let's look at it. Let's look at something like this, okay? This is the trip Operon, right? Now, let's imagine a mutation that would happen in this region right over here, okay? Some mutation in here, okay? Now, what that mutation would do would it probably affect this protein. We're going to call this protein E, right? Whereas a mutation over here would affect this protein, okay? But what about a mutation over here? I mean, this part does not get transcribed into protein, does not get translated, therefore, into protein. So what would happen if there's a mistake here, or here, or here? Now, this is actually, sorry, let me go back. Let's talk about these two first, okay? So these two regions don't become protein. Let me erase that. Actually, that one does become protein. Let's go back to these. Like, these are parts of the switch, so they don't become protein. So what happens if there's a mutation there? Well, if there's a mutation there, what happens is, because that's part of the switch, you're going to be doing something to the switch. You might be breaking the switch for all we know. So for instance, here is a slide of you may have seen this before from last year. These are two birds, okay? One of these is a foot of a chicken, the other one's the foot of a duck. And what you see in the top slide is you're seeing a dye that is used to show you where a protein, and that protein is called BMP4, is being expressed, okay? So these dyes allow us to visualize the protein. You see all this purple? This tells us that BMP4 is working in these regions of this bird's limb, okay? Now, one of the things that BMP4 does is it destroys webbing. So you can imagine, like, for example, imagine your fingers, and when you were in the womb, you actually had webbing between the fingers. All right? Like webbed feet. And what BMP4 does is BMP4 gets rid of it, destroys it. So your fingers look normal. Okay? Now, there is a protein called gremlin. And what gremlin does is it actually stops BMP4. It blocks it. So in other words, if BMP4 removes the webbing, then gremlin would keep the webbing. And that would actually be the difference between a chicken's foot and a duck's foot. If I go back to the slide, you'll notice there's a lot of gremlin expression right here. You can see there's a lot of gremlin in between the fingers, and that stops the webbing from being destroyed. So you end up with webbed feet. Now, why am I showing you this? Because the idea here is that the difference between these 2 feet, the chicken and the duck's feet, is not a mutation in the protein. It's not. It's a mutation in the switch. And what the switch does of a gene is it, well, like we talked about, it controls if the genes are being switched on or switched off, or how much. If it's being switched on and it's being transcribed at a really, really rapid rate. So any mistake in this non-coding section that you can see is involved in regulation affects the regulation of the gene. This is actually what happens in lactose tolerance. So in lactose tolerance, the difference between someone who's lactose tolerant and someone who's lactose intolerant is not because the lactose tolerant individual has a working lactase protein. It's not that at all. The difference is in the regulatory sequence. It's in the switch. In lactose tolerant individuals, the gene switch is always left on. So that individual is always making lactase. In lactose intolerant people, the genes get switched off. So the difference comes down to not the protein, but some mutation in the switch between these two types of individual. Okay? But we'll talk more about mutations the next time we meet. I do want to ask you a question. We talked about mutations in regions that are outside the protein. Here's a question. What would happen if a mutation deleted the repressor gene? What do you think would happen, ladies? Now, in this case, you're deleting the whole gene. So imagine that means you're not going to be producing what? What doesn't get made if the repressor gene is eliminated, deleted? Anybody?
RNA polymerase?
Okay, well, what does the repressor gene, if I look at the slide, what does the repressor gene make?
The RNA.
It does?
Never mind.
No, look at so the repressor gene makes this thing, which becomes this. What's that? That's my repressor protein, right? So the repressor protein comes from the repressor gene. So what happens then if the repressor gene is knocked out? That means no what? No repressor protein. What happens if you have no repressor protein? How would that affect the regulation of the trip operon? Here are your two choices. Either it stays on always or it stays off always. What do you think? Yeah, very good. Stays on. Because if there's no repressor protein, so if you knock out the gene, that means you knock out the protein. Knock out the protein, and there's nothing to attach to the operator region and prevent transcription, which means you're always going to get transcription. You'll get the same effect if we talk about this, because if we knock out this repressor gene here for the Lac Operon, we knock it out, right? Knock out this gene. That means we don't make this protein, which means in this case, the genes for the Lac Operon always get transcribed. Okay. And this, by the way, that's how they actually figure stuff out in genetics, is they do these knockouts where they knock out a gene to see what the effect is. So for instance, in this case, if you knock out the repressor gene and you see that the Lac Operon is always on, then you know that the repressor gene is involved in regulating the Lac Operon. Okay, so again, the big idea is how these proteins regulate genes. Think of them as switches. Think of them as proteins attached to the non-coding part, the non-coding being the part that doesn't become protein, attaching to these regions, the operator, promoter regions, for instance. Even further up, you can see, for example, in the, where is it? The cap, you can see the cap is binding, like you can see it's that section that's not being transcribed and translated, right? Binding there, that offers the ability to regulate these genes. Okay, and that's the big idea here. That's the big idea of how these switches work. Okay, but it's not the only way gene regulation works. Like that's the big idea, but there are a few other things that are really, really important. But any questions about this big idea about gene regulation, about proteins attaching to the non-coding sections like the promoter and operator regions? Like, can you visually see that what's happening? We're okay then? Okay, so then let's look at a few other examples of regulation and then wrap this up, okay? Another thing to consider is, in terms of regulating genes, is the way the chromosomes are wrapped up, okay? So chromosomes are, basically they are, I don't know if you know, I shouldn't assume that, but chromosomes are basically, it's ways of organizing the DNA, right? So the DNA strands are wrapped around these proteins called histones. You can see that here, okay? So what a chromosome is part DNA and part protein. Now, the reason why they're wrapped around the histones is so we can organize the DNA and fit it into the cells. Otherwise, the DNA would just be too long. It's like kind of like a ball of yarn. Otherwise, if you unravel it, it's just too long. It won't fit in the cell. So the chromosomes are a way to organize the DNA so that we can pack them into the nucleus. Okay. Now, if I look at the way they're organized, there's two possibilities. One is called heterochromatin, and the other one's called euchromatin. And the difference between the two is how tightly packed they are. You'll notice that in the euchromatin, it looks like the DNA is exposed. Whereas if I look here at heterochromatin, you'll notice that it's all tightly packed together, right? Very tightly packed. So this is actually important for gene regulation. When the chromosomes are tightly packed in heterochromatin formation, the genes are considered off. And that is kind of logical, because for the gene to be on, it has to be accessible by RNA polymerase, because it has to go in, find a promoter region, start transcribing and translating. But if I look here, is everything's so tightly packed, there's nowhere to get in there and start reading the gene. But if I look over here, you notice that we have exposed DNA, right? Which means something like RNA polymerase has access. If there's a gene sitting in this region right over here, now that RNA polymerase has access to that gene. So the way the chromosomes are organized will also affect gene regulation. It's actually another thing to consider in terms of whether genes are on or off, okay? Again, the big idea is about regulatory proteins, but this is also pretty important too. The second thing on this list, these are other things to consider in terms of gene regulation, is something called methylation. And methylation is when you add a methyl group. So a methyl group is simply, if you remember from your chemistry, when you add a methyl group, you're adding CH3, okay? Now what happens when you add methyl groups to DNA is it actually turns off the genes. In this case, it actually is a way to inactivate an X chromosome. If you recall, females have 2 X chromosomes and males have only 1, okay? Now, This is actually an interesting biological problem because we've been talking about gene regulation, right? Which is the ability to turn on and off genes. Now, where do these genes sit? Well, they sit in strands of DNA on your chromosomes. So what happens if a female has two copies because they have two X's? Each X chromosome carries a copy of a gene. We'll call it gene A, right? Then a female would have two copies of that gene, whereas a male would only have one copy. So we were talking about gene A, and gene A is being transcribed into and translated into protein. Then the problem would be that females would make twice as many molecules of protein A compared to males because they have twice as many copies of the gene. So what happens is that in females, randomly, one of these X chromosomes gets inactivated, gets turned off. And the reason, the method by which that happens is the methyl groups attach, these groups attach to the chromosomes and actually inactivate the chromosome. It's called a bar body, okay? By doing that, even though females have two X chromosomes in every cell, only one of the X chromosomes would be working, and therefore a female cell would express the same amount of protein A compared to a male cell. And if you remember this picture from grade 11, we use this as an example to talk about Calico Cats. And in Calico Cats, they have a gene for coat color, one being black, the other one being orange. So if a female cat randomly shuts off the orange gene on one of her X chromosomes, then part of that cat will be black. Of course, if the female randomly shuts off the black version of coat color on one of the X chromosomes, then the cat will be orange in that part of the cat. Okay, so that's actually another interesting way genes are regulated is through the addition of methyl groups, which actually switch off genes. Okay. Now, the third one is the effects of RNA. But I'm actually going to stop here because I'm actually going to suggest that you watch a video about the role of RNA. And then what we'll do next time is we'll talk about the video that you just watched, okay? It's on something called RNA interference, the role of RNA in actually shutting off genes. Okay, so what I'm going to do is I'm going to stop here because we talked about the big idea in gene regulation, the role of regulatory proteins, and thinking of genes as genetic switches and how they work. And we added a few more examples of gene regulation. But I'm going to stop here and then take questions. Okay, so hold on, let me just stop this.